All right, let's dive into one of the most incredible and essential structures in all of biology, the plasma membrane. It's the gatekeeper, the communicator, and the literal boundary that defines what's alive and what's not. So how in the world does this tiny, tiny barrier pull off such a gigantic job? Let's get into it.
I mean, really think about that for a second. Inside every single one of your cells is this perfectly tuned orchestra of chemical reactions. And outside, well, it's just chaos. So what's standing between that delicate internal order and the complete randomness of the outside world? The answer is this amazing, dynamic barrier.
And when I say tiny, I mean it. This barrier is ridiculously thin. We're talking 7 to 8 nanometers. To put that into perspective for you, a single human hair is about 80,000 nanometers wide. So how can something so impossibly fragile be strong enough and smart enough to run the life of a cell?
Well, its to-do list is just massive. Of course, it separates inside from outside, but it's also constantly exchanging information, controlling every single thing that comes in or goes out, and even lets the cell move, grow, and recognize its neighbors. It's like a security guard, a communications tower, and a shipping depot all rolled into one.
Okay, so to really get how it pulls all this off, we need to look at the architectural blueprint. And the entire thing is built on a really simple but totally ingenious foundation, the phospholipid bilayer.
The whole magic trick starts with a single chemical property. The core molecules, these phospholipids, are what scientists call amphipathic. It's a fancy word, but it just means they have a complete split personality when it comes to water. One part loves it, and the other part, well, it's terrified of it.
And you can see that split personality perfectly right here. Each phospholipid has a hydrophilic head. That part loves water. And then it has 2 hydrophobic tails made of fatty acids. These guys hate water, and they will do absolutely anything to get away from it.
So what happens when you just drop millions of these little molecules into water? They build themselves. It's incredible. The water-fearing tails all dive inward, hiding from the water, while the water-loving heads face outward on both sides. And just like that, poof, you get a stable 2-layered sheet, the phospholipid bilayer. No architect, no instructions needed. It just happens. Pure chemistry.
But you know, a simple barrier, even a self-assembling one, isn't enough. To be truly functional, to do all those jobs we talked about, the membrane needs machinery. And that's where the real power players come in, the proteins.
Now, you might think the membrane is mostly made of those lipids, but check this out. By mass, it's nearly half protein. This isn't just a fatty wall. It's a bustling, busy hub of activity, and it's these proteins that give the membrane most of its specific dynamic functions.
You can see them here just studded throughout that lipid sea. Some of them are called transmembrane proteins, and they span the entire width of the membrane, acting like channels or gates. Then you have others, known as peripheral proteins, that are just attached to one side or the other.
And what do? Oh man, pretty much everything. They're receptors that grab onto hormones and other signals. They're transporters that act like bouncers, deciding which molecules get in or out. Some are enzymes that kickstart chemical reactions right there on the cell's surface, and others act like anchors that connect one cell to another. Each protein is a specialist with a critical job.
Okay, so we've got our lipid foundation and our protein machinery. But there are a couple of other crucial components that add the finishing touches, giving the membrane some refinement and a unique identity.
First up is cholesterol. Now, I know it gets a bad rap sometimes, but in the cell membrane, it is absolutely essential. These molecules just wedge themselves right in between the phospholipids. This brilliant little move stops the lipids from packing too tightly and getting stiff when it's cold, and it also keeps them from getting too loose and soupy when it gets warm. It's basically the membrane's fluidity buffer. And then, on the very outer surface of the cell, you've got these carbohydrate chains hanging off the proteins and lipids. Together, they create this sort of sugary coating called the glycocalyx. Think of this as the cell's ID badge. It's how your immune system and other cells can recognize it as you and not some foreign invader.
So, we've seen all the parts, right? The lipids, the proteins, the cholesterol, the carbs. Now it's time to put it all together into one beautiful, unifying concept. This is it, the fluid-mosaic model. First proposed back in 1972, this idea completely changed how we see the cell membrane. It's not a rigid, static structure. It's a mosaic because of all these different molecules, and it's fluid because they can all move around relative to each other. It's a sea of molecules.
And how fluid are we talking? Well, the phospholipids are constantly on the move, zipping past each other side to side thousands of times a second.
Actually flipping from one layer to the other, what we call a flip-flop, is way, way rarer. That's because it would mean forcing that water-loving head through the oily, water-hating middle. And it just doesn't like to do that.
The degree of fluidity also really depends on the kind of fatty acid tails in those phospholipids. Saturated fats have these nice straight tails that can pack together very neatly, but unsaturated fats, they have kinks or bends in their tails because of some double bond.
And that kink makes all the difference. See how they act like little elbows, pushing their neighbors apart? This stops them from packing in too tightly and makes the whole membrane more fluid, more like olive oil. A membrane packed with saturated fats, on the other hand, is going to be much more viscous. Think more like butter.
So, the membrane is fluid, but why does that matter? Why is this one single property so fundamental that you could basically say fluidity is life?
Because a cell isn't a brick. It has to be dynamic. Fluidity is what lets a cell change its shape, or what lets a white blood cell squeeze through a tiny capillary to fight an infection. Without it, cell division would be totally impossible.
And this property is what enables all those other amazing functions we talked about. Proteins can float around in the membrane to where they're needed, membranes can fuse together to deliver cargo, and the cell can easily grow or repair any damage. It all comes back to being fluid. So what we've seen is that the plasma membrane isn't a static wall at all. It's this carefully orchestrated, fluid dance of molecules. It's a perfect example of how organized chaos makes life possible. And that kind of leaves us with a final thought. What other systems, in biology or even in technology, depend on that exact same balance between structure and dynamic chaos?